this talk will be about female evolution and genomics of female-limited polymorphism through sexual conflict over mating. And it will largely be about my own research organism, which I, without shame, say I really love, but I try to make it more general. And I'm going to argue for the case that female-limited polymorphism, as opposed to, say, male-limited polymorphism, are understudied, but are much more common than we have thought so far. Here are some examples. There will be more later. So phenotypic polymorphisms are organisms where there are two or more discrete or semi-discrete, clearly identified phenotypic clusters within a population that are also heritable to some extent. Some quite famous examples are shown here. To the left, we have the ruff. It's a lekking shorebird with strong sexual selection that occurs in Europe. I believe you can see it here in India on wintering grounds too. It's been known for a long time that there were two male forms, independent territorial males, and which were brownish or dark, and satellite males, which were settling around the territorial males, functioning essentially as some kind of reproductive parasites, trying to get matings from residents. It was then discovered even a few years later, a third morph called feather, which lacks these plumage entirely as the males have and looks exactly like a female. And remarkably, these are they differ not only in color, but in suites of traits, including behavior, aggression, mating success, and other traits. And it was found that this polymorphism comes about through a so-called supergene, which has been created by actually two different inversions generating these three morphs that are maintained by frequency-dependent sexual selection. And another famous bird example are the white-throated sparrow in North America, where you have a white-striped males and females and tan-striped males and females. And the white-striped are, especially the males, are very aggressive, and they are very successful in sexual selection. But they arise from a large chromosomal inversion that spans a large part of the chromosome. But where some of the homozygotes die, but the heterozygotes have quite high success. So these two polymorphisms they occur in females too in these white-throated sparrows, but it's mainly been focused on the sexual selection in these aggressive males. Now, so how do these morphs, discrete morphs, come about? How do we form these clusters within a species, given that recombination is supposed to break down a lot of co-adapted genes or gene complexes? Increasingly, research, including from Krishna Megkunte's lab here in Bangalore, have shown that there can often be genes of major effects, such as generated by various transcription factors, of which the double sex is one quite famous such transcription factor. And Krishna Meg and colleagues showed some years ago that this double sex is very important in four major insect orders, the four big ones, Diptera, Coloptera, Lepidoptera, and Hymenoptera. And it's involved in regulating a lot of remarkable structures, including these impressive beetle horns in sexual selection, where you have males with large horn and males with smaller horns coexisting. But this double sex also is involved in pigmentation, Drosophila melanogaster, in flight adaptation and the formation of wings, and in female-limited polymorphism and mimicry in butterflies. And double sex is interesting because it's also double sex and other genes from the same gene family, DMRT gene family, seems to be involved in female polymorphic systems. such as damselflies that I've been working on, where you have typically a male and then 2 or 3 females, one female being male-like. And these male-like females seem to be masculinized, not only in color, but also in other characters like shape. In Drosophila, other transcription factors have been identified that can turn a light female, which is often thought to be the ancestral female, into a dark female, which developed this black abdomen tip, similar to male. So male-like females occur in several species of Drosophila across the phylogeny. And there can be complex macroevolutionary dynamics, both in damselflies and in Drosophila. So you can have a state where you have sexual dimorphism with male dark abdomen tip and female lacking it, and then it could transcend, move to a state where females get completely looks like males, or it can even go so the males lose the dark tip and females get it, or you could have a situation where none of the sexes have a dark abdomen tip. And the same is true for damselflies. They can have a very dynamic macroevolutionary history with gains and losses of the polymorphisms. So female-limited polymorphisms, as I said, they have been discovered and are increasingly discovered in more and more species. I mentioned insects. I have also worked on diving beetles, where there are females that differ in their elytral structure, so that some females are smooth and others are groovy. And that has to do, it's an adaptation to avoid male clasping and male mating harassment. There is also this wonderful spider in Colombia, I think, with 3 or 4 female polymorphs. I mentioned the butterflies and the Drosophila, where these male-like females also occur. But recently, also fascinating, they have been discovered in vertebrates like birds. Here is an example from South and Central America, where you have hummingbirds. Males look like this, white-necked Jacobin, and female-like females look like this, and then there is a second female morph that looks like males and seem to behave like males, being more territorial. And in European cuckoo, there are also 2 forms, one gray form that looks like males, and one brownish forms that differ. So they seem to be more and more common and increasingly discovered across the animal kingdom. So to understand why they continue to exist, why they are maintained, why do we maintain this polymorphism? We have heard during this course about genetic drift. It's quite a powerful force. When we see variation, we need to explain why it's maintained. To give you a feel for this, I will just make a philosophical and historical excursion here to the French, to the Greek philosopher Aristotle and the French philosopher Buridan, which coined they independently, or whether it was Buridan or Aristotle is still debated, but they formulated the so-called Buridan's ass. And you probably heard about Buridan's ass. The ass, or the donkey, sits here, and there are 2 equally profitable food items. And there is no, for a philosopher, no rational argument for which one it should pick here. And since there is no rational argument, they are both equally profitable, it ends with the donkey starving to death. Some people think this is a stupid, just a stupid philosophical game. Others have spent a lot of time analyzing this. One of the examples where this was possibly had some political consequences were more than 100 years ago in the Senate in the United States. The Senate was very divided, just like Buridan's ass. Where should we build the channel in Central America? should we do it in Nicaragua or Panama? And the Senate was locked in a debate for a long time until they settled on Panama. What has this to do with biology? Well, sexual conflict over mating can promote female polymorphism and force males into this, to what we call a Buridan's ass situation, or a Buridan's ass zone. And this was an idea proposed by Sergei Gavrilets more than 20 years ago. He developed a model for understanding the evolutionary consequences of sexual conflict over mating between males and females, because there is a tension between males wanting to maximize mating rate and females wanted to have a lower optimal mating rates, because matings are often costly physically in terms of time and opportunities. And when Gavrilets formulated this model, there was a lot of focus on sexual conflict leading to speciation. It turned out it seemed like sexual conflict has not been that important in speciation. Empirical data indicate at least. So there was some confusion at this time. When Sergei published a paper, which in my mind is one of his most interesting ones, he suggested that if there is sexual conflict over mating, this could lead, male mating harassment and sexual conflict could lead to the splitting of a population with establishment of 2 or more female genetic clusters, as he would call it, or morphs, I would say. And the reason is that the selection pressure from males force females or selects for diversification in females. And if females are different in looks and phenotypes, males have difficulties in targeting them. So in a way, you can say males function as predators that promote polymorphism in their prey, females in this case. So with this general remarks, I now move over to my study organism, which is the common blue-tailed damselfly, Ischnura elegans, which is an interesting species that occurs from Europe to Japan over the Palearctic. It's interesting because it has 3 female morphs. We see them here. And one of them being a male mimic. She's sitting here in mating with a male, same color. And then one greenish form and one reddish-brownish form. And we have all 3 morphs in most populations we have studied. And the genetics behind this system is very simple. It's a one-locus system with 3 alleles in a dominance hierarchy. So with 3 alleles at one locus, you have 6 possible genotypes, but only 3 of these are phenotypically expressed. If you carry a P allele, then you can be PP, PQ, or PR. All of these will give rise to what we call the androchrome, the A morph, the male mimic, the blue females. So a pure population of androchromes can still carry the alleles of the 2 other morphs in heterozygote states hidden. If you are a QQ and QR, you develop into the I morph, the infuscans morph. And if you're the rarest morph, the O morph, then you're always a homozygote. So it's a very simple genetic system. And data from my group and from other groups, not only on Ischnura elegans, but also on other species, suggest that males have learned or plastic male-mate preference. They can change during their life, and they switch in a population which females they prefer to mate with, or actually which leads to harassment because males are more eager to mate than females. So the males form these plastic search images. When a morph becomes female morph becomes common, she suffers more from male mating harassment in a plastic male plasticity, which leads to a negative frequency dependent selection. Males are also monomorphic. They carry the alleles, but they don't express them. So here is how it works then. So we have monomorphic males with plastic mate preferences. They search for and try to find a female. And then this plasticity affects the fitness of the different females, and this leads to genetic evolutionary dynamics. And here we have some experiments that are not yet published. We look at male-female interactions in relation to the experimentally manipulated frequency, and the blue is the androchrome, the male-like, and you see they encounter more approaches, more contacts, more clasping attempts, and more attempts to form mating through tandem with increasing frequency. And this leads then the change in the female morph composition affects then the males who can switch their mate preferences, and this goes on. So one interesting thing of this plasticity, we wanted to test with a postdoc of mine and Natalie Robertson and a collaborator in the Lund Vision group, was if we can see, are there any signatures of opsin gene expression plasticity in these mate-searching males? Opsin genes has to do, these are genes that occur in both invertebrates and vertebrates, which has to do with the color vision. So we sampled a number of populations with differing morph frequencies in southern Sweden. Almost all populations had all three morphs. The most common is the blue one. It's the male-like. You see it's the majority morph. And then the green one is the I morph. And the red one is the O morph, which we didn't include in this study. So we focused on the two most common ones. And then we measured opsin gene expression in the eyes of the males, because that should be related to how good they are in finding mates. And then we looked at the reflectance of the bodies of these two female morphs in relation to the absorption spectra of the males. And you can see here that the lower one shows the I morph, the female-like morph, and there the male opsin gene absorbance peak coincides quite well with her peak. So they can see her as a potential, identify her. Whereas in the androchrome, the male-like females, they fall in between the two absorbance peaks. So they implying that it's more difficult for males to identify them and supporting that these might be actually function as male mimics. And we also looked at how does opsin gene expression for the different opsin gene of different wavelengths change with the proportion of androchrome females. And there were significant changes, sometimes with curvature, but definitely over the range of more frequencies, males' opsin gene transcription profiles changed, supporting this plasticity scenario. If we look over the long time, we follow a number of populations, around 15, that I followed since the year 2000 when I started this study. It's still ongoing. And this year, 2024, we will sample our 25th generation. They have about one generation a year. Here are for the first 10 years. And here are the frequencies in 12 of these populations. And the blue is the male-like phenotype, and the others are the two other morphs. And you see that they are remarkably stable. The frequencies remain relatively stable. No morph is lost. All populations have all three morphs, although they change. They differ somewhat in the frequency. You can also see here that in some of these populations, it goes up and down, but it seems to return to some long-term equilibrium. And when we analyze these data, it turns out that these morph frequency fluctuation, the morph frequencies are more stable than would expected by drift, strongly suggesting that the negative frequency-dependent selection maintains them at their current level. However, we also see some interesting dynamics happening. Here is one example. This is, let's see if I can find this population here. I started following in 2000. So here we see a trajectory of more frequencies over 20 years or so. I started here in the year 2000 when I just started my first faculty job. And in this ternary plot, each corner means a hypothetical situation of 100% of the morphs, so the population would be fixed, monomorphic. I started in 2000, though male-like females were in majority. And then they have gradually moved down and moved around, and now they are around here, around 60%. So in this population, the androchrome, the male-like females, seem to have decreased. I honestly think I had expected more changes when I started. And we have some data indicating that it's not only sexual conflict, but the morphs also differ in temperature tolerance. And the male-like females, the androchromes, are more common at high latitude. So given that it becomes warmer in Sweden, I would have expected over time or I expect in general that we'll move down here and get even more frequencies. We'll see what the future says. Okay, so we do other things then also. We are also interested in understanding the fitness landscape, what the phenotypic traits of these, how they affect their fitness, and to what extent these morphs, these color morphs, differ in other traits than color. So together with my former postdoc, Moritz Lurig, we have used thousands of images of scanned individuals and in combination with computer vision to calculate a number of different phenotypic parameters from different parts of the body to get an overall view, a multidimensional view of the phenotypes of these. And here are some of the results. So here are two principal components. Here are 4 phenotypes, the male in dark blue, the male-like females, the androchrome in light blue, and the 2 other morphs in yellow and orange respectively. And here we can see that the male mimics, the male-like females, they are closer phenotypically in phenotypic space to the males, although not perfectly overlapping. So you could say they seem to be morphologically and phenotypically male mimics, but imperfect mimics. The 2 other morphs are further away in this morph space. And we can also then plot these phenotypic estimates against mating success, or the probability of finding mating, and generate these fitness surfaces of the different morphs. This is still ongoing. We are adding more data. We also have fecundity data, and we are trying to understand this intraspecific fitness landscape for a quantitative. Yes, Deepa? This one and this one is copulation success. This one is fecundity. We have less for fecundity, unfortunately. Yeah. And fecundity, we put it in females and let them lay eggs. Copulation, mating success is somewhat, is it good for a female to be found in copula or not? So that we are still trying to understand this. But we find some interesting patterns that we are continuing to. So I will just say a little bit also now about the genomics and development of this female color polymorphism. So the larvae, this insect spend time as an aquatic larval stage, and then at the adult stage we have 4 different phenotypes, males and 3 morphs. And here are some interesting natural history details here. So it comes out, emerges, and there are only 2 female forms in the early immature stage. You can also see one interesting aspect here is that the males have this blue patch on the abdomen, and so does the 2 female immature forms. They have a male-like character in the immature stage. Then when they get sexually mature, something interesting happens. The males keep the blue patch, so does the androchrome, the male-like females. But the 2 other non-male-like females, they cover this blue patch with a pigment like melanin. And we have shown that the presence of a blue patch, you can, my former student Beatrice Willing manipulated this by painting it with nail polish of different colors, makes males avoid mating with these. And conversely, if you paint these with blue, they also avoid it. But when females get rid of the blue patch during development, they also become more attractive to males. So it's this particular character, the blue abdomen, functions as a male signal, and it has been exploited by the male-like females. And initially, we looked at this. We looked at female color development during sexual maturation. They all start then. All three female forms start with this blue male patch, and then it's covered in the two other forms with pigment, where it is kept throughout life in the male mimic. Initially, we looked at gene expression profiles, and initially, early stage, there's not much differentiation in terms of which genes are up or down regulated. But as they reach sexual maturity, hundreds of genes are differentially expressed between these morphs. And some of the shared genes, one of those genes was a gene in the DMRT family, the same family as double sex. So initially we thought double sex could be involved, although we, just like in these other systems that Krishna and Kunta worked on, but we didn't have enough, we couldn't make any definitive conclusion there. So more recently, we have sequenced the three female morphs and used a new reference genome resource. So now we have a little bit more understanding of the origin of these female morphs. And it turns out it's quite a complex genomic basis and history. So in the beginning, there was this orange females, the O female, which is the rarest morph in our population. What happened was, we have reasons to think this is the ancestral form. What happens was that there was an inverted duplication giving rise to the male-like female that diverged and through additional duplications. And then the third morph was generated by translocation from this A morph back into the O background. So it was a complicated process involving inversion, duplications, and translocation, resulting in a polymorphic system with three females. If we look in the phylogeny of the genus Ischnura, to which Ischnura elegans belongs, we have 4 possible states where species can be found. We can have female monomorphic, one type of female, which look like males. So female monomorphic with androchrome females, that's a species which is sexually monomorphic with only one type of female, sexually monomorphic and female monomorphic. We can also have a situation where you have female monomorphism, only one type of female, but she looks different from the males. So that's a non-male-like type. And then we have one very common is that you have female polymorphic, 2 female morphs, one being a male mimic. And then you have the rarest one, trimorphic. So 3 female morphs, like in our species, one being a male mimic. And here is a phylogenetic ancestral state reconstruction we did some years ago, where we looked in the phylogeny, presented a new phylogeny for this genus, and looked at what is the evolutionary history. It turns out that the most likely ancestral state was female monomorphic with heterochrome female. Females look different from males. Then female polymorphism, female dimorphism has arisen several times. So a male mimic established itself, and it has happened independently several times. So we went from here to there. And then in some lineages, we have lost the ancestral female and got fixed. So we got female monomorphic for male and similar to males. And then in this particular part of the tree, a third morph established. And it's this part of the tree where our species Ischnura elegans is. So it's only in one part of the tree where we have three morphs. And this was for the genus Ischnura, but this genus belongs to a bigger damselfly family called Synagrinoidea, which are called pond damselflies. And we have now recently finished a manuscript where we look at the presence of female polymorphism across this superfamily. We have data for almost 500 species, phylogenetic data and phenotypic data. And here are the results. This is a global analysis. So polymorphic lineages are red. The polymorphic lineages are mainly two females, sometimes three, but generally two. Sexually dimorphic lineages where females look different from males, but only one type of female, and sexually monomorphic females where females look like males. And we have these states here: sexual dimorphism, only one type of female; sexual monomorphism, one type of females look like males; and female polymorphism. And here we have estimated the transition rates between them, and we find that female polymorphic lineages can give rise to sexually dimorphic lineages by losing the male-like females. They can also give rise to sexually monomorphic lineages where the androchrome, the male-like, is lost. And sexually dimorphic lineages can also give rise to sexually monomorphic lineages. In general, it seems like polymorphic lineages seem to give rise to sexually dimorphic lineages at a higher rate than they're giving rise to sexually monomorphic. And also the origin of female polymorphism is typically sexually dimorphic lineages, and the descendant of female polymorphic is typically sexual dimorphism. So it seems like here along this axis is where most action is taking place. And we have also looked using both fieldwork and habitat data of various ecological factors that could explain the evolution of female polymorphism in these damselflies. So we have done fieldwork in the tropics, collecting data directly, and also compiled data on the literature. So if we look at different latitudes and the habitat, we have classified habitats as tropical or temperate, and the habitat can in turn be closed, that's a forest, or open, that would be an open pond area. So we have 4 types, rough habitat classifications. It turns out that the probability of female polymorphism is higher in temperate areas, and especially high in open areas. So we find polymorphisms in the tropics, but they tend to be in open areas, as shown in yellow. In the temperate zone, female polymorphisms are more common, both in closed and open habitats. Interestingly, we find no effect of operational sex ratio. So there is no operational sex ratio as measured in the field, doesn't predict the occurrence of female polymorphism. But the adult density, as estimated during transects we have done, is a good and significant predictor of female polymorphism. So the probability that the species is polymorphic increases with density of adults, consistent with the sexual conflict, because high density species would be females would be more likely to suffer mating harassment from males. So to summarize this, so female color polymorphism, they are more likely to evolve in open habitats at high latitudes in the temperate zone and when density is high. And here is a little anecdote. This picture is from Cameroon, where we did field work in 2017, and we came to this open area and started catching, and we said, when we started catching here, that this is an open area. Here we predict there will be a damselfly. If there are any damselfly species, they will be polymorphic. We went out and sampled, and correctly we found this sprite. Sorry, forgotten Latin name, but here we have the male and the non-male-like females, and here we found the male-like androchrome females. So we find this was the first time this was found in this species, this polymorphism. And the final thing we have been looking at now in these, using our phylogeny, is that we were interested in what is the role of female color polymorphism, if any, on speciation, extinction, diversification, and lineage turnover. And these analysis, this phylogenetic analysis of diversification, are somewhat controversial. There are difficulties with them, particularly estimating extinction rates, which tend to have very low estimates. So one has to take them with a grain of salt. But given these caveats, we find some interesting patterns. So in general, female polymorphism is associated with higher speciation rates, as shown here in red, compared to the other states. That's sexual dimorphism with only one female that differ from male, or sexual monomorphism, the blue, where males and females look the same. And these are what we call hidden speciation and extinction rates. So we test for at different overall rates, and we find similar patterns across it. Interestingly, higher speciation rates in polymorphic species is also associated with higher extinction rates. So it seems like polymorphic lineages give rise to more species, but it's compensated by these species being short-lived and go extinct at a higher rate. And as a result, the net diversification is not that big, actually, although actually slightly more diversification of polymorphic lineages. But the fact that speciation and extinction are coupled means that the difference becomes less than you would expect. But turnover, that means how fast taxar turnover, is therefore higher for polymorphic lineages. So a more dynamic evolutionary history. To sum up then this, so female phenotypic polymorphism, they are discovered in an increasing number of animal groups. They're particularly common in some insect groups like damselflies and butterflies. One female morph is typically, but not in all systems, it's a male mimic. And they are often maintained by frequency-dependent sexual conflict, although I should say that not all female polymorphic systems are characterized by this, but at least in damselflies and many others. And they can also survive several speciation events, be shared between species, and they can show interesting macroevolutionary dynamics. And with this, I would like to thank my collaborators, my lab, postdocs, PhD students, and others, and the funders, and you for listening. And I end with the same graph as a talk I gave the other day. If you're interested in our work, please contact us. If you're, for instance, want to discuss postdoc opportunities or anything. Thanks for your attention, and I'm happy to answer any questions. Sorry?
The female polymorphisms,
Oh.
they are doing the course of speciation.
Oh.
To retain the polymorphism, or is that not?
Yes, it seems like it seems like it's largely what we call a trans-species polymorphism. So that a bit like MHC or blood groups. So for instance, this genomic study we did, we compared Ischnura senegalensis, which you have here in India, and C. elegans. and the polymorphism is shared between these two species, and they are separated by 5 million years. But of course, that's only 2 species in the phylogeny. We would like to go deeper and look particularly in the North American species, because it could be independent origin also, or hybridization. Sorry? Oh. That's a very good question you ask, because they are. Oh, he asked, is there any variation in the nymphs, the larvae? And I think that's a very interesting question. First of all, there is variation in color among the nymphs. They can range from green to brown to transparent, but I'm not sure that polymorphism is linked at all to the adult polymorphism. So, and the answer to your question is we don't know. We need to genotype the larvae. We just have the tools now to do it. One of the things we would like to do is that even if the color polymorphism is not expressed among the larvae, it could still have pleiotropic effects affecting behavior or aggression. But then we need to identify them, sequence them. So this is the next step. We have collected larvae and we want to look how allele frequencies changed across the life cycle. Did you have a question?
So what are the population sizes there? Because you say drift is not important probably.
Yeah.
So you have migration between populations
we again, as you know, estimating effective population size is a challenge. We have good reason. So we have good reasons to think they are quite big. But in Southern Europe, they are even more stable, apparently. You saw there is little movement in ours, slight movement, but in Southern Europe, they're essentially completely flat. So I think there might be some room for drift that push you away a little bit from the equilibrium. Actually, this has been one of our problems. We would like to have a little bit more disturbance so that that could be more dynamic. Oh. Yeah, that's good. So I think we are getting to how far we can go with comparative methods. I think we have identified 3 factors: density, latitude, and habitat. Density and habitat we can explain from the principle of first principle of sexual conflict. High density, more polymorphism. Open habitat, less ability for females to escape sexual conflict. Latitude, as you know, is one of those. It can mean anything. We have some evidence that the androchrome females are more cold tolerant. So it's not only sexual conflict, it can also be a role for abiotic factor. But again, I think the way I think of this is that it's not temperature or sexual conflict, but both that might interact. And I actually think it's to make one interesting point there is that we believe the male mimicking functions as mimicry. But if you know mimicry theory, it's negative frequency dependent. So the more common the morph is, the less efficient it should be. Up in our populations, uniquely so in Sweden, the androchrome, the male mimic, is 60 to 80%, which suggests that maybe it has increased because of temperature and that mimicry at that high latitude is less efficient. So that was my best hand-waving explanation I could give.
One question on the andromorph.
Oh.
So you said it's kind of successful at avoiding population.
Oh.
And yet one needs to copulate to reach 60-80%.
Exactly. So it's not, this is a very common question. I think essentially all females get mated. So it's more a matter of avoiding the excessive pre-mating harassment. And also remember that if the females reach, say, 99%, that feeds back on the males, right? Because if males would be very picky and not be plastic, they would miss all the mating opportunities. So they will always get some matings.
About the latitude thing, is there a correlation between density and latitude? So maybe these two factors are not independent.
Good question. There is a correlation between latitude and density. So they're not entirely independent, but we have control for it. So but that's true. And as you say it, I find it one of the nice things with doing this field work, doing this rather tedious and boring walking in the forest and doing kind of think, oh, do we really have to do it? But then you look in the literature and people say, oh, it's lower densities in the tropics. And then you look, where is the data? Now we have a great data set of this very descriptive piece of natural history. More people should do that.
All right, Erik is going for a little bit longer. If you think of questions later, catch it. Thank you.
Thank you.